Hello, I am Jorge Velasco, and I'm going to tell you about our experience in the laboratory onion and chick cells, structure, and cell division. We first saw an onion tissue that provided us a good view of the cells. Also, the experiment with the spiral provided us a very good view of the cell structures. We saw some parts of the cells because the microscopes that we used were excellent. What do we need? We need an onion, a microscope, a glass slide, cotton swabs, a cover slip, a Bunsen burner, tweezers, Legal, methylene blue, and alcohol. So the procedure was: first, get 4 of the glasses lights and cover a slip for yourself and make sure they are both thoroughly washed and dried. Second, remove 2 of the single layer of epidermal cells from the inner concave side of the scale leaf, the thinner the better. Third, the epithelium removed leaf it for approximately 30 seconds in alcohol, which will dry the water in the cells. Fourth, remove the epithelium from the alcohol and put one epithelium in the legal and the other in the methylene blue strainers for 15 seconds. Fifth, remove them from the strainers and wash the tissues for 30 seconds. Sixth, place the epithelium you took on each slide. Make sure you don't fold it or wrinkle it. So this is the tissue painted with legal. Those orange dots are the nucleus of the cells. Now this is with methylene blue, but the cells are not visible.
Now let's talk about the cheek lab. First, 2 of the group will offer their cheek tissue. Then use the cotton swabs for remove epithelium from the cheek. Second, place the content extracted in the microscope slide and wait until it dries out completely. Third, go with the professor in order to fix the tissue to the slide with the Bunsen burner. Fourth, apply a drop of 2-methylene blue and legal to each slide for 2 minutes. Fifth, wash it in the sink, but do not apply water directly. Simply let the water run over the slide very gently.
These are the results of the lab. Those orange things are the cells. Here the black things are bubbles and the other things are cells.
First, what is the nucleus for?
The nucleus regulates all cell activity. It is like the boss of a company. It stores the genes of all living systems on chromosomes. Organizing genes onto chromosomes also allows cell separation. Uses nuclear pores to move regulatory factors and gene products. Creates ribosomes in the nucleolus. Produces codes for proteins and organizes the coding of DNA to replicate genes.
Second, what is the nucleolus for?
It's a small body that is inside an eukaryotic cell, composed largely of protein and RNA. In the nucleolus, there are formed the ribosomes when the cell is not under reproduction. The ribosomes are transferred to the nucleus thanks to the pore.
Do the cells you saw divide by what method? Explain why.
The cells that we saw divide by mitosis. Those cells didn't reproduce by meiosis because meiosis is exclusively for sexual cells, and we saw cells from onion tissues, so it's mitosis. Also, the cells from the cheek aren't sexual cells, so they reproduce by mitosis.
Do onions use sexual or asexual reproduction to exist?
Onions reproduce both ways because onions are bulbs, and bulbs are seed plants that produce flowers. So, as other seed plants, the flower must be pollinated in order to form seeds. The pollen is deposited in another bulb, and the reproduction starts just as in a flower. Bulbs are also reproduced asexually with a process called vegetatively reproduction. The bulb splits and creates another bulb named bulblets. Now I'm going to talk to you about my experience in the life in a pond water drop. On the top of the microscope, add one single pond water drop. Take some pictures and make drawings of what you saw. Describe how the organism that you observed moved. Be detailed in your description, include any structures. We saw a Daphnia moving. It moved with the side legs like jumping. With those same legs they breathe because they are primitive animals. We also saw Kilomons that moved like sliding with the help of their tail that we didn't see. We discussed the importance of the microorganisms, described the importance of the microorganisms, and what would happen if all the microorganisms would suddenly disappear for a water pond. Microorganisms create our breathable atmosphere. They make Earth's soil fertile, they drive oceanic life cycles, and extract megatons of greenhouse gases out of the atmosphere every year. The vast majority of biodiversity on this planet exists at the microbial level. This is what we saw. That is a Daphnia. It's one of the microorganisms that we saw. It is the grandfather of the shrimp. Here, at the side of the huge thing, there is a little thing that is a kilomon.